Hi, I'm Jillian Cullen. It is June 15th, 2025, and I'll be talking about Wilson's disease today. I chose this disease based on all the other ones. I think I chose it because I was choosing at work, and I was asking one of the doctors I work with if he'd heard of Wilson's disease or any of the disease we were talking about, and he said yeah, and he's never been able to actually diagnose Wilson's disease. He's never seen it. He's seen symptoms associated with it, but he's never actually diagnosed it, so I thought that was kind of cool to learn about it. So yeah, that's why I chose it. So first, I never really thought about my consumption of copper. At least I didn't before this project, and I know now that we consume copper in our everyday diets. Some foods that do contain copper are avocado, potatoes, dark chocolate, almonds, or kale. Those are just a few of the foods that contain copper. There's lots of others. Those are kind of the main ones I consume, so I thought that was interesting. In a healthy person, this consumption of copper is processed normally throughout the body at the cellular level, although there's a genetic condition, which is called Wilson's disease, that leads to the copper accumulation in the liver and eventually our bloodstream. Well, the bloodstream. And this happens due to a dysfunction in copper metabolism throughout the body, and this dysfunction occurs on the ATP7B gene. This gene encodes a copper transporting ATPase that plays a crucial role in regulating copper homeostasis in the liver. And this little guy here is what the ATP7B gene looks like. I thought it was a little messy guy. I liked him. We're going to take a deeper dive into the etiology behind Wilson's disease. This is where we will look at where the mutation occurs and how this affects the body. The mutation that causes Wilson's disease is on the ATP7B gene, which is located on the 13th chromosome. When there's a mutation on this gene, the mutation that causes Wilson's disease causes this mutation on the ATP7B gene, which means copper is not transported properly in or out of the body, or in or out of our liver cells. And this causes a accumulation of copper in our body. This accumulation is inherited in an autosomal recessive pattern, which means 2 copies of the mutated gene are required to cause the disorder, which means 1 copy from each parent is given to the offspring to give them the disorder. So here we talk about the molecular role of the ATP7B gene and how copper moves throughout our body. Copper is absorbed from the food we eat. It enters the bloodstream, and from there it is delivered to the liver. Copper then enters the liver cells through a transfer protein called CTR1, and then once inside the liver cell, ATP7B gene helps incorporate copper into ceruloplasmin, which is a copper-binding protein that carries copper to tissues in the body via the bloodstream and helps transport the copper in and out of the liver cells. Once copper is incorporated into the ceruloplasmin, ATP7B helps direct excess copper into our vesicles. Once in these vesicles, they transport excess copper to bile to send to the intestines and leave the body through feces. In Wilson's disease, here's a little example. So we have healthy food we eat. It goes to digestion, the digestive tract, and then the bloodstream, and then our liver cells, and then to the Golgi apparatus, then bile to the intestines. But with Wilson's disease, it just goes food we eat, digestive tract, and then it goes to the bloodstream, our liver cells, and it spreads to that's where the accumulation occurs, is in the liver cells, and then it goes to our overflows in the bloodstream and can go to the other organs. And that's where we have issues with when people with Wilson's disease have brain issues or kidney issues. This is caused by excess buildup from just the liver overflowing into our bloodstream and making its way to other organs. Okay, and now we're going to look at the cellular level of Wilson's disease. Here there's a lot of dysfunction when there's a mutation in the ATP7B gene. In a normal functioning liver cell, copper is transported into the Golgi apparatus by ATP7B, that's where in the Golgi apparatus is where copper is turned into ceruloplasmin. With Wilson's disease, that ATP7B gene is failing, and which means it's impairing the change in the Golgi apparatus and no longer being turned into ceruloplasmin and no longer being excreted into bile out of our bodies. And that's where the high accumulation occurs, because if we're not able to, the Golgi apparatus isn't able to do its job and the ATP7B gene isn't able to do its job, then it's just building up copper as we eat it. And then that's where it overflows and goes into the other organs. And that can also build up in the cytosol and the mitochondria of the liver cells, and that's where it starts flowing out. Wilson's disease mainly hurts the liver. It also can spill out of the liver cells into the bloodstream and can reach the brain and kidneys. In the liver, the excess copper causes an activation of reactive oxygen species. These cause oxidative stress and damage parts of the cell. This damage, our reactive oxygen species cause inflammation, fibrosis, and even cell death. And this can cause liver dysfunction and cirrhosis in severe cases of Wilson's disease. Most excess copper also damages the mitochondria, which causes the energy deficits in the liver cells. So the mitochondria is not able to do its job, Golgi apparatus is unable to do its job, and that cell is really dysfunctional and causing a buildup of the copper. Next, we have a buildup in the brain. In the brain, the excess copper accumulates in the basal ganglia, which is where we get most of our motor control from, and in our cortex, which is where we get cognitive function, our ability to think. This accumulation leads to problems with neurotransmitters and signaling, and that can cause symptoms like tremors, like if you see someone's hand shaking, involuntary muscle contractions, and even with really severe cases, there can be some hallucinations. So that's where that cognitive function and our motor control comes into play with those issues. Okay. There is currently no cure for Wilson's disease, although there are two different types of therapy. One is called chelation therapy. This is when there's a drug called penicillamine and tretinoin. They bind to the excess copper ions that aren't being sent to bile, and that allows them to be excreted via the urine rather than through our feces. There's also zinc therapy, which prevents the intestines from absorbing copper, and that reduces the buildup of copper in the liver cells. The last resort for a severely damaged liver, like with cirrhosis or where it is building up too much to go to the brain and to the kidneys, would be a liver transplant. And that would help, a new liver would encourage the normal copper metabolism. The downside of these, besides the liver transplant, the downside of these treatments, the cleating and the zinc, is the cleaning therapy comes with more side effects than the zinc treatment. And the side effects of the cleaning come with side effects that look like neurological system, drug-induced liver damage, some stomach issues, and then also rash. And then there's also issues with the zinc therapy, such as nausea and vomiting, so still some of that GI issues that comes with the cleaning therapy as well, which is interesting. And then there's currently a gene study for patients with Wilson's disease, and the goal of this study would be to deliver a one-time infusion of a normal copy of the ATP7B gene into the liver, and this ideally would aid in the metabolism of copper. And I think they were working through phases of this trial. It was on their website, because it would be a really big deal for patients with Wilson's disease. Sorry, I had to pause it. My mom came downstairs. So the goal of this study would be to do that one-time infusion. It's being done by UltraGenEx, and it's like the gene therapy. And that would be get that ATP7 gene, a normal working ATP7B gene into the liver to aid in the metabolism of copper. This would be a really big deal for patients with Wilson's disease, as it would be a one-time treatment instead of like continually taking either the zinc or the cleating therapy agents for the rest of their lives with those harsh side effects. No one wants to feel nausea and vomiting every day, or it might seem mild, but over and over, and that repeated exposure would make it very hard for those patients. So yeah, thank you for listening. Okay. And then conclusion, there's much more research needed to get that gene therapy actually into motion. I think there's been multiple companies trying to do this gene therapy research. The side effects of the current treatments are causing more issues than actually helping, and because they basically have the same side effects of the actual disease. The liver can take serious damage due to Wilson's disease, can cause Parkinson's-like symptoms, and if the copper accumulates in the brain. Early diagnosis can prevent this severe damage and improve the quality of life for patients, and that early diagnosis can look like genetic testing to help diagnose the chance of having one of those copies in the body. And here's my works cited. Yeah, thanks for listening.